So the brain is highly adept at responding to sensory information. Lights, smells, sounds all come into the brain. But we know also that because social relationships are so important for humans, that social expressions, faces, are highly adept at activating certain brain areas. But we also know that most of the things that we learn about the environment have come from associating colors or stimuli with some kind of emotional response, such as these delicious-looking vegetables. And typically this type of learning theory has it that this happens through classical conditioning. And this is the prototypical example of Pavlovian conditioning, where you have a bell paired with food, and then the animal salivates, and then it salivates in the absence of the food. But you also have something, you can also have aversive learning, where you have to protect yourself from the environment. And in the laboratory, we do this using fear conditioning, where you pair tone with a mild electrical shock to the animal, and the animal learns to be afraid of the tone in the absence of the shock. And it's been shown that there are certain brain regions that if you don't have a fully functioning, intact brain region, such as the amygdala, that you don't have this kind of emotional learning that you make. Cliver-Bucy syndrome is a syndrome where you have damage to the amygdala and people can't form strong emotional memories. So now, what is the neural correlate of this? So it turns out that Hebb, back in the 1970s, he's a neurophysiologist, he came up with this idea of circuits that fire together, wire together. So, sorry, it's going too fast. And we know that the brain is an electrical machine, and we have these neural oscillations that are happening constantly. And it's always been thought that these neural oscillations have been an epiphenomena of the brain, but it turns out that these oscillations are a way that the brain coordinates activity to facilitate types of learning. There are a number of different neuromodulators in the brain. One in particular to emotional learning is acetylcholine. And acetylcholine, specifically, it's been shown that these neurons in the brain oscillate at certain different frequencies. And that these oscillations, that in certain diseases like Alzheimer's disease, these neurons are disrupted. And because of this, this results in cognitive dysfunction, people who can't remember, and there's a steady decline as these patients, the disease progresses. So my research is looking at the interaction between the amygdala and the connectivity of the amygdala and the cholinergic input. So this is just a connectivity matrix of the amygdala. And you could see, like, understanding which of the parts of the brains that are interacting with the amygdala. There's so many different areas, it's hard to kind of pinpoint exactly which are the functional units that kind of facilitate memory. And answers to this problem came from this creature here. This is a green algae. And this algae, because it needs to phototaxic move towards light, scientists have discovered that they have a certain type of channel that's sensitive to light. So if you flash lights onto this channel, you can open up a sodium channel on the left here, which is, in a way, sodium is the way that action potentials are generated in the brain. But you can also activate a chloride channel, which is a way of inactivating neurons. So modern scientists have now found a way to put these different channels into neurons. You put them into a viral construct, and then you can infect neurons with these genes, and the neurons then express these proteins, and then you can control the activity of these neurons with light. And this is just an example of the different types of activity patterns that you can generate. You can have very standardized high-frequency stimulation, but you can also begin to mimic physiologically relevant or natural activity patterns to look for correlations or relationships between activity in the brain and behavior. And that's something that we do. So here, this is some data from our lab where we've infected different cholinergic populations in the brain in transgenic mice, and we looked at projection patterns, colored in green here, into the amygdala. And you could see that there's a very distinct differential input to the different nuclei. So the next thing we wanted to do is try to stimulate these different projections, cholinergic projections, in vivo to see if we can see any kind of effect on amygdala-dependent learning and memory. We first wanted to look at innate behavior, appetitive or aversive behavior in these animals. So what we did is we put animals through this place preference paradigm where they're just free access into this room. And when animals were on one side or the other, we stimulated these different inputs. And what we found, and then on the next day, we allowed them free access, and we saw which area, the chamber they preferred. And we saw that when we stimulated one input, that animals preferred one side, but another input, they avoided it. So we can have these innate types of behaviors. This we supported where we paired a touchscreen box where animals could press a middle square to receive stimulation. And after we paired this a few times, the animals continued to press to get this stimulation, supporting this idea that this is rewarding for the animals to stimulate themselves. So this is just some data where we show that we can kind of generate these kind of oscillations, natural oscillations, by artificially stimulating these fibers. And the idea is then to try to correlate these frequencies with the behavior that we're seeing in the long run. And this is just a power spectrum of the different frequencies that we can generate by simply stimulating these different cholinergic inputs to the amygdala. And I think that's it.
Okay, so I'm definitely not on the science end of this, but are you familiar in Canada with the CCNA project? And I think there's a link to the international piece on the Canadian Consortium for Neurodegeneration and Aging project.
I don't know it, but I'm interested to hear about it.
Well, I'm just curious because there's 20 teams and there's a lot of money being poured into examining neurodegeneration in aging, particularly dementia and Alzheimer's. And I'm on team 20, of course, not on the science side of things, on the social science side. But this seems like this would, be a, incredible connection. And I was
there's a lot of ethical issues. Now, in human groups, so stimulating the brain in humans is kind of a problem. But we do it clinically, but with electrodes. So the traditional one is deep brain stimulation, where you put an electrode, and specifically for Parkinson's diseases, but they're also working for cognitive disorders, such as obsessive compulsive disorder. But these kind of techniques are very non-specific. They just put an electrode in a certain area, but then they stimulate, but you're stimulating everything. But the problem with something like optogenetics is the ethical approval of infecting neurons with non-host genes. So I think within the next 10 years something will happen, but there's still a lot of hurdles in the way of getting it to be...
Yeah, I'm just wondering how far away you are from it.
For me, like with mice, it's not a problem. Yeah. But in terms of the clinical aspect, I would say 5 to 10 years before this is probably being utilized in human groups, clinical groups.
Thank you. Okay, I'm not even going to attempt that. I'm going to do like a handoff.
Very interesting talk. So I'm just wondering, especially in the light of what you just said, 5 to 10 years down the road, so what are the military or intelligence implications for this kind of work?
Yeah, so that's very
fearless kind of soldiers, or intelligence manipulation,
or just controlling behavior with lights. And of course, these are kind of things as scientists we don't think about this until it's too late.
That's what I'm asking now.
Or maybe we should start thinking about it. But yeah, I don't. It's a hard question to kind of answer because. I'm sure that the military, I don't know it very well, but I'm sure they use drugs at the moment to make people feel less fear and be more aggressive. There's a lot of probably also psychological manipulation, obviously, and that's affecting the neurochemistry of the brain. So, in terms of can we, have everyone hooked up to lights and be driving certain kind of crazy behavior, it could be possible, but I guess, first we have to see if we could do it in the mouse. and then we could see if we could do it in a human. But yeah, it's a very interesting question.
Maybe a very simple question. So you seem to indicate at the end that the mouse got a positive experience from the stimulus. Did you also have cases where it led to a negative?
Yeah, so they also, there was one group, depending on the input that you activated, if you stimulated one cholinergic projection to the amygdala, then they preferred that area. But then if you stimulated another, they would avoid it. And so basically, it's kind of showing that the amygdala is receiving this information regarding valence, positive or negative valence. And in a way, then sensory information can come into the amygdala, and these two bits of information can be bound together to make a biophysical change that results in a memory. So that's kind of the work that we're looking at, or doing.